So let's discuss PCR. So there's just some key aspects which I'm going to learn about PCR, which include the steps for PCR and why it happens. So PCR allows you to basically make a whole bunch of DNA which is replicated. And what that allows for is, for example, like fingerprints or gel electrophoresis, you need a lot of it. So in this case, this allows you to get that process of having tons of that same DNA segment and able to analyze it later. So the first step of PCR includes denaturation. So denaturation, basically you have your double strands, right, of DNA, and they are split into single strand because a lot of heat is applied to it, so it allows it to split, and this can allow us to add the new strands on later on. So the second step would then be annealing. So again, you always add annealing. So you always add 3' to 5', and this is where you add the next replicated part of it. 3 elongation, just as it sounds. And the cycle then repeats, making these copies always over and over again. So that's PCR. Yeah, and along with this, we can also talk about reverse transcriptase. So usually in the central dogma of biology, what happens is you go from DNA to RNA to proteins, right? However, there is a method of going backwards. So synthesis of DNA from RNA instead. So in this case, it creates complementary DNA called cDNA, which lacks introns off the mRNA template. And this is naturally often used by viruses. Yeah, so we talked about those two things, and for the next part, we'll talk probably about gel electrophoresis.